Hello and welcome to an overview of Chapter 6, which has everything to do with metabolism and enzymes. So the first thing to know is that when we talk about metabolism, we're really talking about all the chemical reactions within an organism's cells, tissues, and body. And metabolism is just a property that enables life to occur because we're having molecules being exchanged from high to low energy or low to high energy throughout an organism's life. And a metabolic pathway is just a series of steps where we have a molecule as an input, and it ends with a molecule that is some product or end molecule. And each step within a metabolic process is going to be catalyzed or created or started by an enzyme because enzymes have specific shape and structure. And we know that because enzymes are proteins, and proteins fold and form in the endoplasmic reticulum as a result of their DNA sequence. So if we have a metabolic pathway here, each reaction is going to occur getting us to a product, and each enzyme is going to be useful in said product. And we saw this also with cell signaling, where we had a starting molecule or a initial response, and then the cellular product was a response of different enzymes, maybe protein phosphatases, causing that reaction to occur. Now, there's two types of metabolic pathways that are important to know. The first is catabolic. I always think of cannibals within this first type. And catabolic pathways release energy by breaking down very complex molecules into simpler compounds. For example, cellular respiration takes glucose when we have oxygen, and we create ATP as a result. I often think of catabolic pathways as exergonic reactions because heat will be released. And then anabolic, think anabolic steroids, uses energy and time to build up complex molecules from simpler ones. For example, making proteins from amino acids or glycogen from glucose are two types of anabolic pathways. Now, chemical energy is just stored potential energy, like the battery in your phone when it's fully charged, that's available for work during a chemical reaction by being released. And the chemical energy is often stored between the atoms in a molecule, whether they're covalent or ionic. And when those bonds are broken down, energy is released, and it can be converted into other forms of energy. So for example, just to reiterate, here's a person with a lot of potential energy. They're not currently moving. That energy is being released as a person dives because it's now transitioning from stored energy to moving energy. And then a person has to reinvest energy to climb back up to obtain that same potential energy. Good example of an exergonic catabolic reaction and an endergonic anabolic reaction. Now, there's a couple laws of thermodynamics. Many of you might have seen these in chemistry or another science class. But the first is the first law of thermodynamics, which is that energy can only be transferred and transformed. It can't be created or destroyed. Basically, this is the principle of conservation of energy. And a good way of thinking about that is I can't create energy because it is always existing in a system. It's just that at times it may be less usable or more usable. So I go into my house, it's cold, I turn the heat on, propane is combusted, allowing heat to be released. The energy that was stored in the propane tank is now being released as thermal energy, which is going to make the air in my house warmer. But then there's the second law of thermodynamics, which is that every energy transfer or transformation increases the entropy of the universe. And what that means is that during most reactions, energy is lost that becomes unusable and becomes heat. So I have chemical potential energy stored in propane. I turn on the heat. It's combusted, heating up my house. that heat becomes less usable energy. While I can bask in the radiant warmth of my house that's now warm, I can't really recapture and use that heat because it is very low energy, it's not usable, and it can't be used to do any work. So entropy is just a measure of how disorderly or random or low energy a system is. So good example, bear eating some salmon. First law of thermodynamics, the chemical energy is now being released exergonically into the bear's digestive system, which is going to allow the bear to respirate, but also release a lot of it as heat, and that bear can't recapture that heat. It won't be able to use it for future movement or activity. Now, cells create structures from less ordered materials, and same with organisms, and that is a result of using energy to create order and structure, and it takes time and investment. It does not happen automatically. So when we look at tissues or we look at molecules like glycogen or really innate proteins, those aren't happening sporadically. They won't happen without any investment of energy, which is why we need energy to constantly flow into an ecosystem. Now, when we talk about the amount of energy that's available, we often talk about free energy. And free energy is just the energy that can do work when we aren't concerned about temperature and pressure. Now, when we talk about the change in free energy during a chemical reaction, the difference in the free energy has to do with what is the final state and the initial state. And only processes with a negative delta G, meaning the final state is smaller than the initial state, are what we call spontaneous. And spontaneous reactions tend to be catabolic and exergonic. So when the energy level is negative, it means things are generally going from a stable and organized environment to a less organized, less energetic, more entroponic reaction. Spontaneous processes can be harnessed to perform work, such as burning fuel to make a turbine move, such as your car. So a couple of good examples of spontaneous reactions: you climb to the top of a diving board and you spontaneously fall into the lake because of gravity. Diffusion, we have a high concentration of molecules. They will want to move as a result of concentration gradient. There's a change in energy. You have a really high energetic molecule. You break it down into water and CO2. Delta G is also negative because of that energy being released. So in all of these examples where we have higher delta G, or higher stored energy at the beginning and lower energy at the end, we'll have a negative G, change in free energy, because the system has released most of its energy. It's become more stable, but it's also become more random. So that brings us to exergonic and endergonic reactions, which I alluded to earlier. An exergonic reaction just proceeds as a spontaneous reaction, which means delta G is negative, and it generally means that energy is going to be released as heat to the external environment. This generally means that the outside environment will become warmer and also more random. So a good example of that would be maybe you have some toe warmers in your ski boots. Iron oxide reacts with oxygen, releasing energy. That's an exergonic reaction. Alternatively, endergonic reactions absorb energy. They obtain energy, they invest energy, and it's non-spontaneous, meaning it just doesn't happen instantly. G is positive, which means that the final energy product is going to be higher than the initial. The magnitude of delta G is the quantity of energy that's required to drive the reaction. This is sort of like going out and buying things. The amount of money it costs to buy something relates to the amount of time that you might have to work or save or invest to have that thing be obtained. Bigger molecules require bigger changes in G, which means that they are even more non-spontaneous because we need more energy from them. And here's a good visualization of how those two things work, where we have reactants and products, energy either being released or absorbed with a positive or a negative G for exergonic reactions and endergonic reactions. And different systems can have different amounts or different systems. So if we have an isolated system where we have a difference in energy levels. So in this turbine example, we have more stored potential energy in the first turbine or the first holding tank than the second. We have movement. G is going to be negative because energy is being released. But then we reach equilibrium. So an isolated closed system tends to be something that's not good, because when we get to delta G of 0, that's when life ceases. So this is why we always need to have energy flowing into an ecosystem. Once we have a delta G in a cell or an organism or an ecosystem, life stops. An open system means that energy is constantly flowing, which is good because that means that delta G will always be negative. So energy will be being obtained, heat will be being released, things will be able to obtain energy for life. And if we have a multi-step system, this makes the process more efficient because we're theoretically capturing more of that energy as it flows throughout the ecosystem rather than in one fell swoop. This is often why metabolic processes have multiple enzymes and multiple steps, because it's easier to obtain more of the energy as that molecule is broken down. This tends to be less efficient and releases more heat relative to the stored energy in the initial product. There are three main types of work that are used for exergonic reactions: chemical, transport, and mechanical. To do work, cells need to do what's known as energy coupling. And that's because cells can't release a lot of energy during exergonic reactions, otherwise the heat might overwhelm homeostasis. So most energy coupling in cells is a response to producing ATP, which is a small energy currency that cells use because it's efficient and it's effective for their environment. Now, ATP is a molecule that looks quite scary. We have three phosphates, 1, 2, 3, that are bonded to a 5-carbon sugar and a nitrogenous base. And ATP has a lot of stored energy in it because these phosphate groups are negatively charged. And negatively charges like to repel each other. So when a phosphate is hydrolyzed, going from ATP to ADP, energy is released because we've made that molecule go to a more stable, low energy system because that third phosphate was very unstable. And that means if we want to go from ADP to ATP, we're going to have to invest some energy from an exergonic reaction. The bonds between the phosphate groups are hydrolyzed, being broken apart, polymers and monomers. That's part of chapter 3. Energy is released, and that allows us to do work. Now, ATP drives endergonic reactions by phosphorylation. So when we take that third phosphate molecule and we phosphorylate something like an enzyme or a protein, those molecules obtain that energy stored in that phosphate. The recipient molecule that we often call that is a phosphorylated intermediate. And it's called an intermediate because it's not going to be permanently activated or heightened in terms of its energy. It's going to have a specific job that uses that phosphate temporarily, and once that job has been occurred, the phosphate is going to detach. And this causes a constant cycle of ATP being used, and that phosphate being recycled to go to ADP to form ATP. And this often is important because the ATP phosphate causes a change in the protein shape, which means it allows it to change its shape, maybe allowing it to bind to another molecule, allowing it to move. Many things happen as a result of that phosphate change. And we can see that with things like transport proteins, making our concentration gradient go from low to high against what diffusion would like, and things like transport vesicles and motor proteins that use ATP to make the protein actually move on the microtubules. And this is how we can have an energy coupling between ATP and water being exergonically broken down to ADP, allowing us to do work, but then we're releasing energy from things like breaking down glucose, which allows ADP and a phosphate to be endergonically built back up to ATP. So it's this never-ending cycle of exergonic and endergonic reactions, where an exergonic reaction will fuel an endergonic reaction, and an exergonic or an endergonic reaction requires some level of exergonic activity. Which brings us to enzymes. So enzymes are a way in which cells can obtain more efficient energy transformations, lowering that delta G, which means that less work is required or more work isn't lost to heat. And it's because enzymes are a catalyst, meaning it's a chemical agent that speeds up reactions, and it doesn't get consumed by the reaction, so the enzyme is reusable time and time again. For example, we can break apart a disaccharide, sucrose, with the enzyme sucrose, adding a water molecule to get a glucose and a fructose molecule. And remember, this is called hydrolysis, when we add a water molecule with an enzyme to split a larger molecule into smaller molecules. Now, every chemical reaction that occurs that either builds up or releases energy, exergonic or endergonic, means that molecules have to have their bonds broken and those bonds reformed. The initial energy to start that chemical reaction, starting to make the chemical bonds break between a molecule such as sucrose, is called the activation energy. It's the amount of energy that we have to invest to sort of make the molecule a wee bit unstable to get it to break apart. This activation energy is often supplied in the form of thermal energy, because as you make molecules heat up, those molecules tend to become less stable, and those bonds are easier to break apart. So we can have two molecules, A, B, C, D. We heat them up, these bonds become weaker, and then we have a release of energy as those bonds are broken and reformed to AC and BD. An interesting thing to note is that delta G is negative here, so this is an exergonic reaction. And that means that the molecule AC and BD have less stored energy, are more entroponic than A, B, and CD. So this would be like going from a large protein or carbohydrate to simpler molecules like water and CO2. Now, enzymes make the reaction occur faster, and they also lower the activation energy, which means that we can invest less energy. The amount of delta G is unaffected by the enzyme, meaning what we started with and what we finished doesn't change, but this energy is what we have to invest to first get it. So theoretically, we're making our reactions more efficient in the net term. Now, the reactant that an enzyme acts on is called a substrate, and that enzyme binds to a very specific part of the enzyme that we call the active site. And this temporary bind, sort of like a ligand, is called an enzyme substrate complex. The enzyme specifically then uses that fit and that form to actively manipulate the substrate's structure, physically weakening the chemical bonds that exist between the atoms in the substrate. And with that changed shape, the molecule begins to break apart, allowing us to then release energy for that exergonic reaction to take place. And the induced fit model of the enzyme to the substrate is just the idea that different chemical groups of the active site sort of enable the reaction to catalyze very easily, because the molecule fits itself into the enzyme, making it change its shape as a result of having that external environment around it. It's sort of like if you put your fist into your other hand, your hand then sort of changes shape as a result of having your other hand as a fist within it. We can sort of see how this would work, where the substrate flows into the active site, and then as a result, both molecules start to change shape, which would then allow the exergonic reaction to take place. The active site can lower an EA barrier, as I've mentioned before, by orienting the substrate correctly, straining substrate bonds, providing a favorable microenvironment, so maybe the acidity or the temperature might be different than the external environment, and also covalently bonding to the substrate, so actually tearing apart the molecule by becoming part of it. The substrate's going to enter the enzyme complex. This is where we might have covalent bonds form, and then the products are released, and this is where we would see the change in delta G. And then we now have the enzyme available for the next reaction to take place. Now, a couple of things can really affect an enzyme's activity. General environmental factors such as temperature and pH can make an enzyme work more efficiently or less efficiently. And there's also chemicals that can specifically influence whether the enzyme is considered functional or not. So, for example, we have the optimal temperature for a human enzyme and the enzymes of a thermophilic or heat-tolerant bacteria. They live in things like the hot springs of Yellowstone. Clearly, humans would not function well in those hot springs due to the change in temperature and vice versa. And then if you look in your body, pepsin, an enzyme that breaks apart protein molecules, loves the very acidic environment of your stomach, while trypsin is far more of a basic enzyme that prefers the small intestine. Now, a couple of things can also modify enzyme productivity. Cofactors are non-protein enzyme helpers that could be something like a vitamin B or a mineral, and they tend to bind to enzymes and help them become more efficient, or they bind to the substrates and allow them to have a higher affinity. So this is why having a balanced diet is essential. These cofactors often make enzymes work more efficiently. Competitive inhibitors bind to the active site of an enzyme, and they often compete with the substrate. Competitive inhibitors are often used in environments where the substrate levels are low, so we want to preserve that quantity by turning off the enzymes. A non-competitive inhibitor does the same exact product or result, but it often binds to another part of the enzyme, causing it to change shape and making it less effective. And these are all examples of things that can cause inhibition of enzymes. Remembering chapter 42 and bioaccumulation, toxins, poisons, and pesticides in the environment can make animals die because of their enzymes being turned off. Here's a good example of competitive inhibition and non-competitive inhibition. Another thing that can happen is allosteric regulation that can either inhibit or stimulate an enzyme's activity. And this occurs when another type of molecule, a regulatory molecule, binds to a protein at one site and it affects the protein's function at another site. So for example, we can have an allosteric enzyme with 4 different subunits, and an activator binds to a part of the protein and it becomes stabilized as an active form, which allows for the substrate to then sort of come in and activate. Another thing that can happen is one substrate can bind to one part of the protein, and then the other parts of the enzyme as a result turn on. And then we can also have inhibition where the molecule binds to the protein and causes it to not have any binds occur, which means chemical reactions would cease. And then we also can have feedback inhibition in enzymes. So in feedback inhibition, that means the end product of a pathway shuts down the pathway's initial response. And this is a really useful thing for cells not overdoing a reaction or not synthesizing more products that are needed. So this is a good way to minimize excessive exergonic and endergonic reactions. And a good example would be the end product of isoleucine being a feedback inhibition. It's going to allosterically bind to the enzyme tyrosine, and it prevents this molecule from binding to it and having this chemical reaction occur. And we often see this in molecules such as mitochondria, or I should say organelles such as mitochondria, where it's very important that cells regulate the amount of energy being released and broken down as a result of energy demands, but also available energy sources. So your mitochondria might transition from using glucose to fatty acids to amino acids to make ATP based on the availability of these different molecules and their concentrations.